Well, good morning, everyone. And for some of you on the other part of the half of the world, good afternoon. And I wanted to wish you a warm welcome to our Precisionary Scientific webinar series. I'm Abby, and I serve as a Chief Scientific Officer here, and I'm very happy to have all of you join us. And it's a real joy to introduce today's speaker, Dr. Thijs Verhoog from the Joseph Raimondo Laboratory at the University of Cape Town. So Thijs has devoted his career to a wonderful question: how our very human thoughts arise from the electrical chatter of our neurons. And he does it in one of the most remarkable ways there is, by studying living human brain tissue itself. So today he's going to share how his team keeps that tissue alive to listen in on real human neuronal communication and what it reveals about how our brains grow up and develop. So it's really close to our work here at Precisionary and our support of neuronal research, particularly electrophysiology. So Dr. Verhoog, thank you for joining us all the way from Cape Town, and over to you.
Thanks for the introduction, Abby. It's a pleasure to be here. I've seen a couple of these webinars myself, and it's really nice to now be able to present myself. So yeah, the work I want to show you is how we use this surgically resected human brain tissue to study the physiological maturation of human neurons. And so this is some of the work that we've been doing here at the University of Cape Town and at the Neuroscience Institute. So to start, just a little overview of human brain maturation. So you will be aware that humans have this extraordinary extended period of brain development. It's really one of the defining features of the human brain that it takes so long to develop. And if you look at other species, we seem to be a bit of an extreme example of the general rule that the larger the brain you have and the higher the cognitive capacities that you need later in life, the longer the brain takes to develop. So there must be some very interesting and important things happening during this longer period of development, processes that take time and that allow you to develop these higher cognitive functions. Now, from anatomical studies, we know quite a bit of what is happening already. You see, a lot of this brain maturation is happening after birth. So you may be born with a brain volume of around 25% of the full brain volume, and in the years that follow, lots of things are happening. You get these neurons some of them are still migrating. Neurons start to grow their dendrites and axons. And synapses are produced to form circuits. And then on a sort of a longer time scale, you get the myelinization of long fiber bundles to facilitate the long-range communication. And of course, the process of synaptic pruning to really refine your circuitries. Now, crucially, all of these processes are really stretched over long periods of time, like months to years. And interestingly, the pace of this maturation seems to be in part governed from the bottom up. There have been some recent studies that really show this very nicely, that the genes that are involved in maturing the brain and that are involved in maturing neurons and circuits are actually packed away behind these epigenetic barriers that limit their access and thereby slow down the pace of maturation. So there really seems to be an intentional slowness to this development. And so you can imagine, like, the genes that are involved in the outgrowth of the neuron, those are difficult to reach, and therefore their expression is sort of paced. Now, interestingly, if you take away these barriers, the maturation speeds up. And this doesn't apply only to like structural things relating to the anatomy of cells, but also extends to their physiology. And indeed, if you take away these barriers, in this study that came out in Nature 2 years ago. They showed that in cultured cells, the maturation of physiological properties would also speed up. So basically, it seems that anatomy and physiology both is intentionally slow in human neurons due to these epigenetic barriers. So that raises the interesting question as to what may be happening on the neuronal side, right? We've seen all these very slow anatomical changes. What is happening in terms of the neuronal electrical properties and the functional maturation? What kind of time scale do they mature? So that leads to the main questions of like, what are these trajectories of physiological maturation in the human brain? And how do these relate to overall brain development? And then finally, how does that compare to mouse, which is the model species choice in most of neuroscience and on which most of our understanding is based? But mice have a completely different developmental strategy. Their development, their brain development, is really geared towards fast maturation. So in many ways, they are on the opposite end of the spectrum. Now, the reason that these questions are still open is very practical. And that is that to study the electrophysiology of neurons, they need to be alive. So post-mortem tissue or fixed tissue will not work. And that results in a situation that pretty much everything that we know about the electrical properties of brain cells, how neurons process and store information, and how they are shaped during brain maturation, that is all, that full understanding, it comes from studying the brains of laboratory animals, which are typically rats and mice, a few weeks old. Now we can learn a tremendous amount from these rodent studies, but at some point, I think you'll agree, we must see to what extent those findings will translate to the human brain, particularly given their completely different rates of maturation. And the fact that most of these studies are done on like 2-week-old mice, that's really the sweet spot in terms of brain slice electrophysiology, and that is for a very practical reason as well. That is the age in which slices are the most healthy in mice and where patching is easiest. But what is a P14 mouse and how developed is it, and how does that compare to like the human development? What would be the human equivalent age? That is quite an open question. So to tackle such problems, recently or over the past decade, more and more labs have started turning to local neurosurgeons to obtain surgical specimens and study the physiology of neurons in those. So at neurosurgery, this is often epilepsy or tumor cases. There will be cases where there is an epileptic focus or a tumor in a deeper region in the brain, often for instance in the temporal lobe, and where a piece of cortex needs to be removed just to gain access to that deeper region. So that sort of cortical access tissue is reasonably normal. It's considered to be, well, as normal as we can get. And that suddenly opens up this possibility of studying live brain tissue. So just to give you a little overview of how that works in the lab in practice, this is a little image of one of our specimens. You can see it's a little gyrus, sort of curving around there. There's some damage there due to the surgical procedure of burning the blood vessels to make sure that the bleeding stops. But in here is this beautiful undamaged piece of cortex that holds living cells. Now the challenge is to keep these cells alive, and at the moment they are cut off from the blood supply, so they will be starved of oxygen. So what we want to do is we want to make these very thin slices and bathe those slices in an oxygenated artificial brain fluid so that the cells stay alive. But there's a challenge to that, because the neurons that we want to record from, the beautiful pyramidal neurons that sit in the cortex here, they have a very specific orientation. They all radiate out to the cortical surface. And that's fine if the cortical surface is straight, but there's often turns and twists that it goes through. And if we would cut sections across, or like not perpendicular to the cortical surface, we would be cutting through all those cells, and that would greatly affect the viability of the cells. So we must find a bit of cortical surface that's straight so that we can nicely cut these parallel sections that will preserve most of the dendritic architecture and therewith keep our cells happy. So that's the challenge. In this a little resection here, I would go for that bit there. You see straight edge, cut that out, and then we can put it into one of the, because we use the compressome from Precisionary, and here we see the resulting bit of cortex in the specimen tube embedded in this low melting point agar, and then we can mount it for slicing in the compressome. This is a different block, but you can see how nicely we can have the whole gyrus sitting in there and then cut successive sections from that block. Just a little explanation as to why we're using a compressome here. In my previous lab, we were often using the vibratomes, but the vibratomes, they don't, the technique we used for mounting the tissue there did not involve having this agar embedding. So the tissue would sit slightly loose, and it would be very prone to pieces of pia, which sit on the top of the cortex, blocking the cutting of the blade, and therefore cutting skew slices or even pushing the specimen off of its mount. So that's what I like about this compressome technique, that you really hold the slice from all angles in one spot so that you can neatly cut through. And it also decreases the burden or the time that you need to spend removing the pia here. That's a very tough material, right? It sits on the top of the cortex, and it has all these blood vessels in there. And particularly those blood vessels can be very tough to cut through. So in this orientation, yeah, using the compressor, we don't have to spend as much time on that as we did on the vibratomes. So that gives us these beautiful acute slices, which we can then start to do our experiments on. This is my rig here in Cape Town. There's a lot going on, but the idea is very simple. You've got this microscope here, and underneath the objective there is a little bath where we can pop the slice in, and there it will be perfused with some nice warm HCSF at body temperature, oxygenated to provide the oxygen. And then using these manipulators, we can lower these robotic arms onto which we can navigate a little glass patch pipette onto individual cell bodies. So this is the tip of a glass pipette, which is about 1 to 2 micrometers wide at the tip, and basically suction onto the cell, make a little hole, and then we have a little contact with the inside of the cell, so we can start engaging with it electrically and start stimulating and see how it responds. So that's basically the pipeline of how we use surgical specimens to interrogate the physiology of human neurons. So with that setup, we made use of the fact that we've got 2 hospitals here in Cape Town in the close vicinity that regularly perform operations on patients, ranging from 3 months to 80 years old. So like the full lifespan is available. And the data that I want to show you here is from a data set of 80 subjects covering like the developmental period from 9 months on, or what is it, 10 months on to like middle age, of exclusively temporal cortex resections. Same slicing procedures I just showed you, and then we've got these patch clamp recordings where we inject all these different stimuli and see how the cells behave electrically. So this colorful ring here is just a little overview of our data set, spanning from infancy to adulthood. We are recording, each dot is a single recording, a single cell that we recorded, and we're recording across the layers 2 and 3, so the supragranular layers, really sampling across the layers, across depth and across ages. So that allows us to then characterize the developmental trajectories of human neurophysiology. I'm not really sure about my audience. I just thought I'd introduce the patch clamp technique very briefly. It's very simple. We've got this little electrode here, which is filled with the solution that mimics the internal of the cell. And we have got this electrical access to the interior of the cell. And then we can record, then we can inject currents via this pipette. And then we can measure the voltage response to those currents. So it's pretty straightforward in that sense. So for instance, you can inject a hyperpolarizing current like this, and then you can measure the corresponding voltage change. And then you can start calculating electrical properties. For instance, using Ohm's law, we can take the amplitude of the current injection and the amplitude of the voltage response to get a measure of the resistance of the cell, so the resistance of the membrane. Or we can look at, we can fit sort of the time it takes for the membrane to charge. You can see the voltage response is not instantaneous. It has a little curvature to it, and the time constant of that curvature is also is a product of the resistance and the capacitance of the cell. So we can really start to interrogate the basic properties of these neurons. And when we started to do that for our human data set, we found something rather surprising. And that is that, so these are some experiments we do exactly this for different ages. And you can see that 10-month-old has a much greater voltage response to this hyperpolarizing current injection than a 40-year-old. It's almost a half the amplitude. So this is our data. Each dot is again a single recording, and this is a GLM fit to that data. And as you can see, there is this big change from infancy to adulthood, from 100 megaohm resistance to half that in adulthood. And more importantly, this change takes place over decades. So not like maybe by the second or third decade, that's only when you actually reach adult levels. And the same was true for the membrane time constant. So that thing that we fit here, that also halved over the same sort of time course. And these are really basic electrical properties that govern basically how much a neuron will respond to input and the time course that it does so. That's very, very basic features of how neurons will integrate inputs. Another feature was the voltage sag. You can see that if in a 5-year-old, the voltage response to a hyperpolarizing current injection is rather flat, but in adults, there seems to be some sort of rebound. We call that the sag voltage, and it is indicative of the expression of a specific channel, a hyperpolarization activated cation channel to be precise. And the reason why that's interesting is because that channel determines at what frequency neurons will resonate. So this is an indicator that as you develop and as you mature, the preferred input frequencies of neurons may change, and that this is something that is only sort of really established in adulthood. So extremely slow changes in very, very basic properties of these cells. This also extends to more concepts like the excitability of neurons, like the amount of current you need to generate an action potential also increases with age very gradually. And finally, action potential stability is also something that seems to undergo a lot of change over our development and also only reaches adult levels very late. So what you see is that juvenile cells are in a train of action potentials. When a cell is firing repeatedly, juvenile cells will struggle to maintain the shape of that action potential. They show a lot of use-dependent change, and the action potential broadens quite considerably. And that is not the case in adults. So that leads us to conclude that these human neurons show a very protracted physiological development that continues well into adulthood. Another thing that I found quite interesting is that if you look here, you see there's quite a lot of spread in the data. Now, part of that is due to our sampling strategy, that we're sampling across depths when we're recording. But even if you account for that, there remains a lot of variance, and that this variance is, for instance, for input resistance, changes with development. So you see a lot of variance in infancy, and that is not because individual subjects are different from each other. This is within subject variance. So it gives you the situation that within an individual infant, you may have a very, very juvenile physiology in terms of resistance, interspersed with cells that have very mature physiology. So there's like a very wide range of resistances in the network. And you see these changes in variance for many of the features. And then we wanted to know how this compares to what we know from mouse. So we collected a mouse data set, which shows pretty much the same changes. I mean, the magnitude of the change is very different, but the nature of the change is very similar. It's just that the time course, as you can see, this is years, whereas this is days. The time course is very, very different. So now, obviously, you're probably not surprised by that. We already know that the human brain is very slow to mature, and the mouse brain very fast. So we next wanted to know, like, how fast or like how slow is the human brain? Is it in proportion? So we started to look at the developmental trajectories in relation to brain volume growth. So this is for mouse. All these different colors represent different electrical features that we quantified, and then we min-max normalized the curves, and then overlaid on that the brain volume curve. And you can see that many features closely track the maturation of the physiological features closely track brain volume growth. So it's very much in step. Whereas in humans, that was not the case. This is the human brain growth curve, and you can see that physiology lags behind quite considerably. So mouse physiological maturation closely tracks brain growth, but the human timeline is really much protracted. So to put a number to this, we next wanted to, we did some dynamic time warp analysis, where basically we take the two brain volume curves, and what you can do with this dynamic time warping is see how much you must warp one time signal to become the other. And what you get is this warping path, which is this gray curve here, which will map mouse days onto human years. And what we found was that from around 60% brain volume onwards, which is the warping path was pretty much linear, which means that from that point onwards, the human and mouse brain development, the rate of development is scaled by a single constant, which in this case was 162 days. So the brain volume proportion that a mouse gains in a day takes a human 162 days. So that's a very handy number that we can now take to the physiology and see how that scales. And what we found, we did this for all the variables. I'm just going to show you two examples. For instance, for neuronal resistance, we get a much higher scaling factor, and memory time constant the same, a much higher scaling factor, meaning that it is much slower than you would predict just from differences in brain growth rate alone. And so that's we found that it was anything between 2 and 6 times slower than you would expect after accounting for different anatomical growth rates.
Sorry to interrupt, Thijs. It seems we have a question from one of the attendees.
Sure. How can I?
Anirudh Kohali,
Yeah.
I believe. Go ahead.
Yes, sir. Yes, sir. I have a question here that there are few of the scientists, even they are trying to put the iPSC, that is the induced pluripotent stem cells, of mouse. I'm sorry, of the humans that they put into the mouse brain in order to see the neuronal activity. So, I just wanted to understand that is it something actually related to what you are speaking right now?
Yeah, that literature is relevant, I would say. It's relevant to the point I made in the introduction, in the sense that the pace of neural maturation seems to be governed from the bottom up. It seems to be a cell intrinsic thing. Because even if you graft these human cells into the mouse, the human cells still develop quite slowly. So they really have this, it's not like external factors that govern it's not entirely external factors that govern the rate of maturation. It seems that human neurons really have something within themselves that make them mature very slowly. And so that may be related to these epigenetic factors. I know that people growing organoids also
yeah, absolutely,
have to deal with the fact that human and other non-human primates,
non-human primates, correct, like marmosets or macaws.
Yeah, typically have much slower maturation rates than if you make the same organoids from rodents.
Okay, thank you, sir. Thank you so much.
You're welcome. So the next thing we turned our attention to is to see if there may be something different in the allometric scaling rules that apply. So just to explain this concept, across biology, there's a lot of things often scale together predictably, and one of the most well-known one is how body mass and brain mass scale together, and this is across many species, and it just forms a straight line in log space. So these follow power laws, so that's a cross-species allometric rule. And interestingly, these allometric rules also extend to physiology. It was recently shown in a sort of a cross-species study of the electrophysiology of many different species, that there were these scaling rules applied to the neurophysiology of neurons, where the total surface area of the cell and the total potassium conductance followed these allometric scaling rules. So yeah, there's much. I'm not really doing justice to this paper. There's a lot more to that paper, particularly also showing that humans may be a little bit of an oddball in that relationship. But interesting. But the question we had is whether such allometric scaling rules could also be at work in our developmental data, because that is often also the case that as you grow. There are often these allometric rules that govern the relative scaling of different parts of the body, but also perhaps for neurophysiology. But the latter has never really been shown for mice or for any physiological neuronal property, I believe. So we looked at our data on logarithmic scales and found that mice have this very strong relationship between physiological properties and brain volume. So this is only looking at data during the growth phase of the brain. And you see that there are very strong linear relationships in this log-log space where with strong correlations. And this was much less the case in humans. So you see much weaker correlations and much shallower slopes. So, yeah, it seems that there is a very strong allometric relationship between the maturation of physiological features and brain volume in mouse, but that in humans, this coupling is much less pronounced. And that may be part of why human neurons take much longer to develop. So the last sort of domain I want to touch on is looking at cell type diversity in the human cortex. In recent years, there's been quite some work, mostly pioneered by the Allen Brain Institute, showing how the supragranular layers of the temporal cortex are very diverse, lots of different cell types. And we wanted to know whether this diversity, how this diversity comes about, basically. So what we did is we took all of our physiological features that we measured for a subset of like 400 neurons, where we had all those 25 features, and did some sort of clustering analysis. This is a UMAP. For those not familiar with it, it's basically taking those 25 features and collapsing that onto a 2D plane, where cells that are more similar to each other will end up closer together. And as you can see, if you color the cells by developmental age category, you can see there's a progression through this space, from infancy being in the lower left, all the way to adulthood in the upper right. And if you do some sort of Gaussian mixture modeling, we ended up with 5 E-types, or I should probably say E-states. The first 4 are basically subdividing this sort of this continuum here. But then this 5th type is really separate, and really forms a separate cluster. And interestingly, that 5th type only has adult cells in it. So if you look at the proportion of these different cells over development, you see that this orange cluster only appears in adulthood. And so it really seems to be a late emerging physiological phenotype. So if you do some slingshot trajectory analysis through this sort of multidimensional physiological space, if you will, that'll give you 2 putative maturation paths from infancy to this E-type 5, and then the other one ends up in this other adult endpoint. And interestingly, the key thing that distinguishes these 2 endpoints is their firing properties. So if you get these cells to fire, cells that end up here are the ones that start any train of action potentials with a short high frequency burst, whereas the other ones do not. So this is coloring those cells by their endpoint, and you can see that the bursting cells, these stars, only appear in adolescence, and so that drives this sort of physiological specialization. Now they also differ in other features. For instance, they have a more pronounced sag ratio. So that's that feature that I showed you in one of the first slides, which I said influenced their resonance properties. So indeed, this type here shows a much greater resonant frequency than the other E-types. And what you can see, what happened here, that one of these green cells, the E-type 4, will if you inject a chirp, which is basically a sine wave with increasing frequency, you'll see that the maximum response will be, you'll see that the voltage response is greatest at, for instance, 1 or 2 hertz, and then becomes smaller and smaller, whereas in other cells, the maximum response is at the lowest frequencies, like half a hertz or 1 hertz. So it's a shift in like what kind of inputs these cells respond to most. And so they've got this burst firing, they've got strong resonance, and they're also located slightly deeper in the layers. So that together makes them very similar to the deep transcriptomic cell types that have been defined in layer 2-3 by the Allen Brain Institute, which constitute the classes of cells that are intratelencephalic, so they're projection neurons. And in that sense, the burst firing is actually quite interesting. Indeed, for projection neurons, the burst firing may be very useful to more faithfully transmit information over longer distances. So that would mean that basically the distinct physiology, which has been commented on before, of the neurons in the deeper layers of the supragranular cortex, so that's in these deeper layers, that characteristic physiology is only something that emerges late in development. And it's also interesting that this is absent in mice, right? Mice do not have this bursting at all. So it's a specialization that I believe will probably be shared by other primates, because primate studies which show like layer 2-3 neurons bursting, and also showing this higher sag ratios. So that concludes our results. Just want to wrap it up with some conclusions. And that I think shown that the nature of the postnatal changes in physiology is conserved between humans and mice, but that the time scale is vastly different. These human neurons really mature slower, even if you account for the differences in growth rates compared to mice. And what that ultimately means is that the different stages of cognitive development really rely on functionally different neurons, which I thought was an interesting conclusion. So with that, I want to thank you for your attention and acknowledge there's a lot of people that are always involved in these human studies, obviously on the University of Cape Town side and my previous laboratory in Amsterdam, where we've used some of the data. But first and foremost, of course, the people from Red Cross Children's Hospital and the Mediclinics here, who have been incredibly supportive of this work and who've made it all possible to actually access this extremely rare pediatric tissue and allowed us to investigate these exciting things. So thanks for your attention.
Great. Thank you very much, Dr. Verhoog. At this point, we'll love to open it up to any additional questions or comments. So yeah, feel free.
Yeah, actually, we just wanted to actually request you that if you can please record this webinar and send it to us. Okay, so that we can also do some sort of knowledge sharing amongst our MARSAP colleagues in India. Okay, and we can have a better understanding of the lecture that we had today, Mr. Matt.
Certainly. Yeah. So the webinar is currently being recorded. We will be posting it on our company's website as well as our YouTube channel after the event wraps up.
Okay. Thank you so much. Thank you so much.
Of course. Of course. Any other questions? Okay. Well, if there's nothing else, thank you so much, Dr. Verhoog, for your great talk. Very interesting, obviously. And if anything else comes along, happy to, like I said, we'll be sharing it on our YouTube channel, so you can certainly comment on there for additional questions. And Dr. Verhoog, I'm not sure if you'd be open to soliciting questions via email or additional comments, but we can certainly do that as well.
Sure, yeah. If time permits, I'll definitely be open to answering any questions. Yeah.
Terrific. Well, thank you, everybody. Appreciate everyone joining today. All right. That's it. Thank you.